Okay, thanks, Misha. Also, thanks for the invitation. So, as you can tell from my slides, I'm right now at Boehringer Ingelheim, but I've only been there for about a month, actually. And all the things I'm going to talk about today are from my time at the Vienna Biocenter core facilities, which is the core facility where also Arthur, who just spoke, is working. And this is our group there. So we're going to take a little bit of a step back now. So Arthur told you how you can evaluate a sample once you have it, but now I want to explain to you how you can get a sample of a protein complex. And if you want to express a complex or work with a complex, there's different approaches you can take. So the first approach actually doesn't involve recombinant expression at all, but some stable complexes can actually be isolated from native sources. So some really well-known examples of this are, of course, ribosomes. So many of the ribosome structures available are from native ribosomes, isolated from cells. Also, the RNA polymerase structure was first solved this way. But of course, probably most of the complexes that you're interested, this isolation isn't possible because they're not expressed in high enough amounts. So then there's other approaches you can take. So one is that you express all the single proteins of the complex you're interested in alone, and then mix them together and try to reconstitute the complex. So this can work in some cases, but often proteins that are in a very stable, large complex won't be stable on their own, so you may not be able to express them at all. And then you have to go for a co-expression approach. And there are different systems for multi-gene expression. I'm going to tell you about some of them. And there's also another approach that you can take, which is called a polyprotein approach, which I'll also describe. So what do you have to think about if you want to express a protein complex, especially if it's a rather large one? So first, you're going to have to somehow make a construct that contains all of your proteins. So you need to have an efficient molecular cloning system. So for some expression systems, these are already available. For others, it's not as straightforward. But for E. coli, which is, of course, one of the most commonly used systems, there are systems, and here you can easily express up to 8 proteins at a time. For the insect cell system, so the baculovirus-based system, there are different systems available where you can actually go up to 25 proteins. And for mammalian cells, actually here the approach normally taken is co-transfection of proteins expressed on different plasmids. So here the limitation is really how many different DNA plasmids can you get into your mammalian cell. And I can't really tell you what the limit is. There was a recent paper where they used at least 10 different plasmids and could co-transfect them. So we'll have to see what the limits really are. Another thing to keep in mind is that once you have a construct, probably this is not going to be the final construct that will give you the best results. So you're going to have to screen multiple different constructs and sort of go through this process a few times. So you may have to have different compositions, so add some subunits, take away others. You might have to have some subunits present in higher copy numbers because maybe they just tend to express lower than all the other subunits, and then you'll have a homogeneity issue. You also might need to change your purification strategy. So be prepared that these steps are going to take actually quite some time until you really have your optimal construct for cryo-EM or structural studies. Another important point, and also getting back to what Arthur was discussing, is you need some way to evaluate that what you're getting from your expression and purification trials is really a useful complex. So there's different techniques you can use for this. Arthur mentioned some, so mass spectrometry, mass photometry, proteoplex are some that you're going to hear about later this afternoon. There's many others as well. So I'm not going to talk about this any further, but just to keep in mind that you need some way to evaluate that what you're doing makes sense and is going to get you the result that you want. So here's just an outline of some of the most common expression systems that are available and that are used. So in bacteria, of course, the most common system we have is E. coli. Yeast, the most common yeast used is Pichia pastoris. In insect cell expression, you can do expression also in Drosophila S2 cells, but the most commonly used system involves generation of a baculovirus, which can be used to express SF9 or TNE cells. And then for mammalian cell systems, the most commonly used ones are either HEK or CHO cells. So advantages and disadvantages, so probably everyone is pretty familiar with E. coli, so it's quite easy to use, it's quite cheap, but if you have a very large complex or a very, like a big eukaryotic complex, it's unlikely that you'll get this to work in E. coli. For yeast, it's also quite easy to use. The big advantage of Pichia is really for secretion. Here, I'm actually not so familiar with multi-gene expression systems that would be available in yeast. For this yeast, you normally have to integrate your plasmid stably into the genome, so this is a bit difficult if you have to integrate many genes. So I'm not really aware of systems for Pichia. For insect cells, this is really commonly used for protein complex expression. So you can get very high yields from insect cells. There are established systems. The only disadvantage is that it's a bit slow because you first usually have this generation step where you have to make a baculovirus. You also need some specialized equipment. For HEK cells, of course, if you have specific post-translational modifications you need, and you're working on a mammalian complex, then you most likely have to go to HEK cells. And as I already mentioned here, the advantage is that you can co-transfect many different plasmids, so you don't have to worry too much about getting all the proteins onto one construct. Expression in HEKs is, of course, quite expensive, and the yields that you get are pretty low. But for cryo-EM, that may not be such a big issue. So another thing, once you've thought about which system you need to use, another thing you should think about is what tags you're going to use. So if you want to purify a complex, it's probably not a good idea to use really big, bulky solubility tags like MBP or GST. So you probably mostly want to stick with the small tags, which I'm sure many of you are familiar with. So I'm sure everyone knows the hist tags, so either 6 or 10 histidines that you can attach to your protein, bind to a nickel column, and then elute with imidazole. So this is really commonly used. Another short tag that's used quite often is the strep2 tag, or it's sometimes used doubly, and then it's called the twin strep tag. And this is an artificial sequence that was developed to bind to a modified form of streptavidin, which is called streptactin. So you can buy a streptactin resin, and then you add a strep tag to your protein, and then you can elute your protein with a modified form of biotin called desthiobiatin. So this is a quite specific interaction, so you normally have less background binding than with a nickel tag, his tag. It tends to be a little bit more expensive, and at least in our hands in the facility, we usually saw that if you had an N-terminal strep tag versus a his tag, there you had slightly lower expression levels. Then there's one other tag, which is the flag tag. So this is also an artificial sequence that was developed, and then the capture of the tag is done using a flag antibody that's coupled to beads, and then you elute with the peptide sequence alone. This also gives this is a really specific interaction. It gives really nice purification. So out of all three of these, this one for sure will give you the cleanest purification. The only disadvantage is that because it's an antibody-based system, the flag resin tends to be very expensive, and it's a little bit more difficult to regenerate it than the other systems. But if cost is not an issue, then certainly the flag tag is a really good choice. And usually for complexes, what people often do is not just use one tag on one subunit, but perhaps tag 2 different subunits with different tags, so you can then do basically a double affinity pull down, or a double 2 steps in a row. Another way that affinity tags are usually used is for a system called tap tagging, which was initially, I think, developed in yeast. So it was often used to isolate native complexes. So you would add this tag on the C-terminus of an endogenously expressed protein. You can, of course, also do this recombinantly, overexpress one of the complex components recombinantly with such a tag. And then this is also essentially to do a tandem affinity purification with 2 affinity steps. So the original tag was using the calmodulin binding peptide and protein A. So protein A binds to IgG beads. and the calmodulin binding peptide binds to calmodulin, but you can do this with any two affinity tags. And the idea is that you would have one subunit with two tags, and then the other components of the complex would bind, and then you can isolate the really specific binding partners away from the contaminants by having these two steps in series. Of course, here you have washing steps in between and so forth, so if you have transient complexes or even some subunits that are only transiently associated, then having such a dual strategy might be a bit too harsh. And that's one of the things that you would just have to optimize depending on your specific protein complex. And again, this is something that you may have to make additional constructs and play around with tags and try to figure out which system is really going to work best for your complex, or even which subunit you should tag. So now once you've thought about what system you want to use and what tag you want to use, now you actually have to make your construct. And now because we're talking about multi-gene expression, this is not as straightforward as simply cloning a gene into a vector. So for E. coli, in principle, you can just have different genes on multiple plasmids. The only thing that you have to be aware of is that plasmids, E. coli plasmids, have different classes of origins of replication. And these origins basically determine how that plasmid is replicated inside the E. coli cell. And if you have plasmids with the same type of origin, also the copy number of those plasmids is, let's say, co-regulated. So if you have two plasmids that have the same type of origin and they're normally present at around 20 copies, you'll end up with only, in best case, you'll have 10 copies of one and 10 copies of the other, because the replication of those plasmids is coupled. And this can sometimes lead to that if you try to do co-expression with two plasmids that have the same origin, you may eventually lose one of the plasmids, or one of the plasmids will be present in far more copies than the other. So to get around this problem, there's a system that's been around for a long time, actually. It's called PET-Duet. This is a series of 5 vectors that was initially sold by Novogen, which has now been bought by some other company. I think it's now available through Thermo Fisher. These are 5 vectors where 2 genes can be cloned into each of these vectors, and these vectors all have compatible origins of replication and different antibiotic resistances. So these are the 5 vectors and the origins that are available. So these origins are all replicated by different types of machinery in the cell. So in principle, the replication of these plasmids should not compete with each other. And then you can select for the presence of all plasmids in your bacteria by simply using a combination of different antibiotics. There are 5 vectors in the system, but you can only combine 4. So these last 2 are basically, one is a high copy version and one is a low copy version. So you can, the maximum you can do is 4, but that means that the maximum you can do is 8. There are some other systems available where you can also put more than 2 genes in a single vector, but in principle there's not too many complexes that have more than 8 subunits that you can really overexpress easily in E. coli. So for larger complexes, one usually uses a eukaryotic expression system. So this is just an example of using the PET-Duet system that it's a complex we purified in the facility that had 3 proteins. One of the genes was cloned into the PET vector. It had an N-terminal His tag. And the 2 other genes were cloned into the PCDF duet vector, so they were compatible and had different resistances. Then we just ran a nickel column, cut off the His tag, did ion exchange and size exclusion. And this was a really stable complex, so you can see that even after this purification protocol, we really had a nice homogeneous complex with all 3 proteins present. And if we then ran the size exclusion profile, I don't know if you can see this, probably in the back because of the light color, but basically you get one nice homogeneous peak on size exclusion as well. So the system for smaller complexes really works well. So now if you want to go to a eukaryotic expression system, then things get a little bit more complex. So first I just want to tell you, so I'm only going to talk about the insect cell system, so the baculovirus system. just for lack of time. But I'll first just briefly tell you, for those of you who aren't familiar with the system, how this works. So this system is based first on generating a baculovirus, which then you can amplify, and then you use this baculovirus to scale up expression in larger amounts. So first, to generate such a virus, you first have to clone your gene into a so-called transfer plasmid. So this transfer plasmid, you have your gene, your expression cassette is flanked by two transposase sites, so TN7 transposase, and then you transform this plasmid containing your gene into a special strain of bacteria that contains the whole virus backbone to generate the backmid that you're then going to use to generate virus. So this bacterial cell also expresses this transposase, and you get insertion of your expression cassette into the backbone of the virus. You can then grow the bacteria and purify the backmid, and now you have the whole viral genome. Then you can take that DNA that represents the viral genome, and you can then transfect it into insect cells. And then you wait for a few days, and then you will produce your first stock of virus, which is usually called V0. And then you might have to do one or more amplification steps to get enough virus to then infect a larger amount of insect cells to get enough material for testing. And just so you can see this backmid in more detail. So there's a few years ago, there was a backmid developed by Imre Berger's lab, which is called MBAC-Y. And what they did is they also integrated a copy of the YFP protein into the backmid, so that now when you perform this virus generation step, you can monitor virus production through fluorescence by looking for fluorescence of YFP. And here on another site in the baculovirus genome, you have this TN7 transposition site where you would integrate your plasmid. So this system that I just described is called the back-to-back system. This is the most commonly used baculovirus system. There is another system that you might hear about, which is called flashback. And in that system, you don't perform this transposition into the backmid in this bacterial cell. Instead, you have a linearized backmid, and you have a plasmid that has some sequences from the backmid flanking your gene, and then you actually transfect both of these into insect cells, and by homologous recombination, you get integration of your gene. But in principle, this is the only difference is this last step, how you generate the virus. Both systems are working perfectly fine. So for insect cells, there are several systems that exist for complex generation. So I'm going to mention a few of them, and then I'm going to describe one of them in more detail. So in principle, the first strategy that you can use is that you could simply take each of your genes of interest and make separate viruses that express each of those genes, and then you can co-infect cells with these different baculoviruses. And this is what was done before there were any multi-gene systems available. This is difficult because it's difficult to make sure that that each cell is really infected by all viruses, so it's difficult to control stoichiometry. And also, this really only works up until you have maybe 3 viruses or so, and if you have to use 4, 5, or 6 viruses, then this gets really complicated to really get a really homogeneous complex. So the first system that was developed is called the multibac system. This was also developed by Imre Berger, who made this MBAC-Y backmid. And this system was based on Cre-Lox recombination, so you would clone each of your genes into a plasmid that has a LOX-P site, and then you would do a in vitro Cre-Lox recombination, where you would combine the whole plasmid, 2 to 3 or up to 4, I think, whole plasmids into one huge single large plasmid, and then you could use this to generate a baculovirus. The only problem with this system was that it was very difficult to control the copy number of each plasmid, so you had to make sure that you only have really one set of these transposition sites in your construct, otherwise you won't get transfer of all of your subunits into your virus. So you had to do this by carefully checking how the assembly actually happened and how many copies of each plasmid you have there. So that made it a little bit tedious. Then a few years ago, there was a system developed called BigBack, and this was developed by Florian Weissmann, who was at that time in Jan Michael Peters' group at the IMP in Vienna. And this system is based on Gibson assembly, which is also a form of basically recombination-based cloning. So what he did is he designed different overhangs, so a set of 5 different overhangs. And he would add these overhangs onto each of his genes by PCR. So here, for example, I hope everyone can see this, but here are basically 5 genes. He adds during PCR certain overhangs onto each gene so that they can only recombine in a certain order. So gene 1 has overhangs alpha beta, gene 2 has beta gamma, and so forth. And then by performing a Gibson assembly reaction, he then gets a final plasmid that has 5 expression cassettes in order. And this system works quite well, and you can make up to 25 genes in a single insect cell expression vector with this system. The efficiency is about 30 to 50%. And so when we wanted to take this over and use it in the facility, for us, this efficiency wasn't really high enough just because of cost and efficiency. So we actually developed an additional system, which is based on Golden Gate cloning. So this is a very similar system in that we're going to take expression cassettes and stick them together, but rather than Gibson assembly, it uses a different type of cloning that's based on type 2S restriction enzymes. And these are enzymes that cut outside of the recognition sequence. So here's the recognition sequence for the enzyme BSA1, but actually when it cuts, it leaves an overhang here of 4 base pairs. And the sequence of those 4 base pairs doesn't matter. You can make that sequence into whatever you like. And then when you cut with this enzyme, you end up with one piece of DNA that has the BSA1 site, and one site that has an overhang but no BSA1 site remaining. So the advantage is that you can then go through several cycles of restriction and ligation, and you will always eliminate the site on one piece of DNA, and you'll be able to enrich for your final product. So just to illustrate that a bit better. So if you have, for example, here, your gene of interest, flanked by Golden Gate sites, and then here you have a vector where you have inverse sites. Now when you cut this insert and this vector with the same enzyme at the same time, you're going to end up with this insert with these specific overhangs and this vector with these specific overhangs. And in the end, you're going to get a final construct where you removed all of the BSA1 sites. And because you can, in principle, design the overhangs here to have whatever sequence you like, this system lends itself perfectly to multi-gene assembly. So this is probably too small for you to see, but I have a slide after this that shows it in a bit closer up. But in principle, how the system works is we designed 15 vectors that all have different overhangs that are compatible with each other. So from 1 to 15, you can combine them all in a specific order. So you would first clone your genes of interest into one of these vectors with the correct overhangs, using whatever cloning method you like. And then you'll get a series of entry vectors where you have now an expression cassette for one gene. And this plasmid can also be used just to express that gene on its own. But what you can also do then is take this plasmid and combine it with other plasmids that have the compatible overhangs, and then run a Golden Gate reaction with a final destination vector, and you'll end up with a plasmid where you have all of the expression cassettes inserted in a specific order. And then you can use this to generate a baculovirus. So just to illustrate this in a bit more detail, so if here we have 5 genes that we want to combine, we clone them first into the vectors that will give them these specific overhangs. So here we have these overhangs match, these match, these match, and these match. So they can only combine in a certain order, and you can only get one copy of each gene. And then when you add your destination vector, you're going to get all of these 5 expression cassettes combined into this one vector, like this. So we wanted to test how this system works. So I mentioned before Florian Weissmann, who developed the Big Bag system. He had previously developed a strategy for expressing the APC, so the anaphase promoting complex, or also called the cyclosome. And he did this with his Big Bag system, and then we wanted to also check if this would work with our Golden Bag system. So he developed a strategy where he basically made 2 viruses. So the APC has 14 genes. Some of them are present then as, some of the proteins are present as dimers. So in total you have 20 proteins, and the whole thing is about 1.5 megadaltons. And he made one virus that contains all 14 genes, and then he had to make a second virus that contained only 6 of those genes that form a subcomplex, and this was really necessary to get a nice homogeneous complex. He then had a 3-step purification protocol that he performed. So here, I just show you, this also might be difficult to see, but here is just, these are the DNA constructs that we made. So here is the full construct containing all 14 genes in one copy. So we did here a restriction digest to verify that all fragments were in. You probably can't see this from the back. And then we also made the smaller, what he calls the platform construct that contains 6 genes. We then made those 2 viruses, co-infected insect cells with them, and then Florian purified the complex. And here what you see is a Coomassie stain gel showing that indeed you get all 14 of the different proteins expressed. And what was really nice about this is that Florian then was also able to use this recombinant APC to determine a cryo-EM structure, and he was able to go to a resolution of 7 angstroms. And before that, the material used for studying the APC had been isolated from HeLa cells, so it was basically the native APC complex, but they were only able to get 20 angstrom resolution. So here you can see that really optimizing your expression and trying to get a recombinant form of your complex can really help you get better data. So there's one more thing that I wanted to mention, which is the polyprotein approach. And here what you do, instead of having each individual expression cassettes for each of your genes, you link those open reading frames together with protease cleavage sites in between. Then you co-express the protease, and it will cleave all of the proteins, and then in the end you can form your complex. And this is basically a strategy stolen from viruses. This is typically how viruses express their proteins. And this can be used to improve subunit stoichiometry. So here I just show you an example from a publication. So here they made first an expression plasmid where they had 3 genes expressed under their own promoters in a single plasmid. And you can see here, this is the purification that they were getting of those 3 proteins. So you can see that 2 of them were present in high amounts, but this one was really low. So what they did then is they designed a construct with 3 proteins co-expressed with the TEV protease, and in between the proteins was a TEV protease cleavage site. So from expressing them all from one promoter, you would then get a polyprotein that was then cut by the protease, and you can see then they were able to get a much more homogeneous complex. So this is a really nice approach, but it's a bit tricky, of course, because you have to design this really carefully to make sure that this cleavage is really going to work. But if you have a problem like this, this might be an approach that you can take. So there was one last thing I wanted to say, and that's if you've been trying to express a complex and it's really not working and you want to give up, don't give up. Don't be afraid to change what you're doing. So there's one example we have from the facility of a small, only heterodimeric complex where a student tried to express this in E. coli for about 6 months because it was published that it worked, but she really got nowhere. She couldn't reproduce the results, and then she finally decided to turn to insect cells. And then the insect cell expression, you can see this was really nice. This is just elution from a nickel column. She got something like 40 mg per liter of this complex. So just by changing, the expression system she was using. So be aware that you might have a long road ahead of you if you want to get a really nice homogeneous complex, and you're going to have to try a lot of different things. And with that, I will stop. And I just want to acknowledge everyone at the VBCF, especially Jana, who was the main developer of the Golden Bag system. Also Florian, who helped us a lot with that, and Sonia, who let me show her gel in the previous slide. So thanks, and I'll take any questions.